Hello, welcome. Understanding the chemical nature and chemical nature of all the interactions and reactions within the cell, and especially those that are involved with the maintenance of the information as a genome, and also the expression of the genome is everything. After all, biology is on the foundations of chemistry. So we'll try to have a brief discussion about the chemistry of how splicing happens. So as we discussed, we have an exon and an exon, and somehow we need to get rid of the intron that is present between these two exons. We splice the exons. And we have also discussed about how the branch A site actually plays a role in doing that. The 2' OH would get activated and then will make a nucleophilic attack at the 5' splice site, that is here. And then there is a formation of a lariat. And there is a second transesterification reaction where the 3' OH of the upstream exon would bind here, making a complete exon-exon junctions, that is 2 exons together. And then the intron lariat is removed, and which will then be degraded. So the chemistry wise, it's for a closer look at the chemistry of how and what happens. If you remember, we have discussed about all the nucleic acids on the backbone. They would add 5' to 3' OH, or 5' to 3' linkage. That is the foundation for all of the backbone of the nucleic acids. Here we find a different type of linkage, which is called as 2' to 5' linkage. We'll just have a look at the chemical structure of what the lariat would look like. So this is the adenine, that is the center of what we are trying to look at. This is the adenine, that is the branch site adenine that is present, which carries out the first transesterification reaction. Upon activation, the 2' OH would make a nucleophilic attack and bind to the 5' splice site of the intron. So what we are representing here, everything shown in say purple, is the lariat. So the 2' OH, which would have been this part, that is, we will just have a little bit of saying this is intron 1, just say, okay, let's write up some things. This would have been 1' carbon, and this would be the 2' carbon. This is the 3', 4th and 5' carbon, and then we have the phosphate backbone there. The 2' OH, which was this, would get activated and bind to this would have been on the guanine present on the exon intron junctions, and this is on the 5' splice site. So the 2' OH is binding to the 5' phosphate of the guanine that is present on the 5' splice site. And as usual, the adenine, the branch site adenine, is bound to the other its regular place where it was before, that is this one. Adenine would have had a phosphodiester bond on either side, and then now it has made another linkage using the 2' OH and binding to the guanine of the 5' splice site, and that is this. So if we say there are two different types of linkages that are present here, one of it is the say 5' 3' linkage, but you can call it the reverse way, 3' 5' linkage as well. And now we have a new one, that is 2' to 5' linkage, because we call it this is the 2' and it is linking with the 5th carbon of the guanine here, so we call it as 2' 5' linkage. This is the third type of linkage that we have come across. If you remember, during transcription, a G cap is added to the 5' side of the mRNA, and there the linkage is 5' to 5' linkage. And so this is the third type of linkage that we are learning in terms of the with regard to nucleic acids. So the lariat, once it has formed, this is also called as the three-way junction, because you find that there are three different ways that you can see. This is one way, this is another, and this is another. And in the center of it is this branch site adenine. Thank you very much. We'll see you again.